be of assistance?
All is good. I believe he is connecting now, actually.
Okay. Anything I can do to be of assistance?
I believe I'm all good. Thank you, Doctor.
Awesome.
Daniel, just one quick request while we're kind of waiting for everybody to log on. If we would love to get a picture of you to post on the Honors College Facebook.
Mm-hmm.
So if you want to send a picture to Chip and he can send it to me, or you can send it directly to me.
Okay, I have it, so I'll send that right to you afterwards.
Yeah, that's perfect. A lot of people have been sending like a picture of them holding their title page or something like that. Whatever you feel represents you to the world, would be glad to have it. So
sounds great. I'll make sure to write that down right now so I don't forget.
Yeah. Thanks.
Thank you.
Yeah.
Are you getting on for Daniel's thesis, question mark? Check your email for a link.
You guys make sure you're muted.
Daniel, are we missing anybody? Are we about ready?
Yeah, we can get going. I'm going to share my screen. Oh, is someone recording already?
Looks like someone is recording. It says recording.
Okay, awesome. Well, whoever's recording, if you wouldn't mind sharing that with me afterwards, that would be fantastic. I would appreciate that.
So Daniel, let me just give a quick intro. So
Thank you.
before you get started here, so my name is Jeremy Farrell, and I've been working with Daniel this past semester, chairing the thesis, and I'm really excited for the work that he's done and the opportunity to present to everybody. And so this is the final step of completion for his undergraduate honors thesis, and this is the presentation that accompanies a research paper that's also been completed. So Daniel, please take it away.
Excellent. Thank you, Dr. Farrell. I appreciate it. So also, thank you everyone for logging in today and coming to my virtual thesis. I guess definitely an upside is that I'm able to have some family and friends here, which I really appreciate you all coming. So my thesis looked at using compost as a nutrient source in deep water culture hydroponics. So first off, a little background on hydroponics. Hydroponics is the process of growing plants without a soil medium. And this technology has existed since the Hanging Gardens of Babylon. However, in 1937, it was modernized when researchers began adding nutrients into the water that plants were growing in. And this allowed plants to grow much longer and to be able to grow plants that are much more energy intensive. So that's when we first started seeing crops being used for this. Here, there's two diagrams of different hydroponic methods. This one here is really common, especially in commercial growing, where water is pumped out of a nutrient solution and down a reservoir, and then it's recycled back in. The one that's used in this experience, and that's very common with most home growers, is the deep water culture system, which is where plants are placed directly into a nutrient solution, and their roots have access to that constantly, and it's aerated so the roots consistently receive oxygen. So on the right is a picture of a commercial hydroponic farm. Some advantages of hydroponics is that it reduces logistical issues associated with food production, as this no longer needs to happen in an open field with arable soil, but can happen indoors in warehouses, inside schools, so on and so forth. These plants grow at 2 times the rate as traditional agriculture, since the roots don't have to fight the soil in order to get nutrients. They're more readily available. This helps reduce price volatility and shortages, as these crops are less susceptible to weather changes and blights that may occur in open soil. They also require less space than traditional agriculture and have a significant reduction in water and energy usage, given that you don't have heavy machinery and that the water is consistently recycled throughout the system. So all plants, regardless of whether or not they're grown in traditional agriculture or in hydroponics, require the same nutrients to live. That's a chart on the right of that. However, the most important thing that we need to look at, and that we'll reference later, is this NPK ratio here. And what this is your key nutrients that the plant needs to be able to survive. So every single fertilizer that you buy, any hydroponic nutrient chemical formula that you buy will have an NPK ratio on it. And that'll change based on what plant you're trying to grow. So some issues with traditional nutrient solutions. This is a chart that breaks down what soil-based growing goes through and the nutrient flow through those systems, and in hydroponics. So the hydroponic growing, as you can see, significant amount of steps are removed. However, what's not included in this chart is the actual manufacturing, refining, packaging, and transportation of these chemical nutrients that we use in hydroponic systems. And that's often left out of the energy equation. And as a result, once we include that back in, sometimes hydroponics really is comparable to traditional agriculture in terms of energy consumption. So in this thesis experiment, we'll be looking if we can leach nutrients from compost in order to feed plants grown hydroponically. So our research questions will be, how does using compost to feed hydroponically grown plants compare to using a traditional chemical nutrient? Will plants fed with compost grow at the same rate as traditional hydroponics? And does living bacteria and fungi in compost help hydroponic plants grow? That's a really important question that we're looking at here, because bacteria, protozoa, and fungi, all things that are naturally occurring inside soil and in compost, are really beneficial to plant growth and help promote a very healthy root ecosystem. However, when we're using traditional hydroponics with the chemical nutrients, we're getting rid of all that bacteria, fungi, and protozoa. So by using the compost leaching and introducing those healthy bacteria, fungi, and protozoa, it's possible that that could help the plants grow. So can we use compost as a nutrient source? This is what your traditional flow chart throughout hydroponics or throughout compost is going to look like, where we're importing raw materials, and those are often things like food scraps, lawn clippings, mulch, things of the sort, putting those in a concentrated area, and then our finished material, we have microorganisms and those raw nutrients that are much more readily available. So the plants grown in this experiment were basil. And as you can see here, every plant started out in a seeding tray inside these rock wool cubes, which are extruded or superheated and extruded rock. It creates it's like a cotton candy-like substance. So every plant was seeded in the rock wool cubes and in the humidity dome before being moved into the hydroponic system. This is what your traditional immature or young basil plants are going to look like. One thing that's important is that before you transplant into the system, you ensure that you have pairs of leaves in its young stage, because that's an excellent sign of plant health. So the experimental setup, we had 9 totes, and a tote is one of these large growing systems that we use in hydroponics. And we had 9 of those. 3 were used for the hydroponic standard traditional growing methods with a nutrient or chemical solution. 1 was with sterilized compost, and 1 was grown with non-sterilized compost. All of these had a 50-watt resistive heater in there so that we could maintain temperature at about 71 to 73 degrees, as well as an air stone that provided 1 liter of oxygen a minute. The control totes received 7 tablespoons of MaxiGrow, which is a very commonly used hydroponic nutrient, both at the home grower and for commercial operations. The totes that were aerated or that had the compost solution, the compost was placed in 1 gallon cheesecloth bags, tied off, and then placed in the water and aerated for 24 hours using the air stone. The ones that were compost were placed in an Instant Pot and cooked or pressure cooked for 25 minutes at about 250 degrees. This is what the whole system looked like. This did take place in my apartment. It was quite crammed for a little bit, but it was really great being able to just take measurements here at home, especially given what had been going on. So this is a picture of the setup. In this front row here, we have our controls, followed by variable 1, which was the sterilized compost. In the back row was variable 2, or non-sterilized compost. The lights were run off of this timer for 11 hours a day, which is about what Basil wants. So preparing the compost that we collected was from Appalachian State University, their physical plant and dining services. The compost, when we initially collected it, was at 130.2 degrees Fahrenheit, and that's both a pro and a con for us. That high temperature indicates very good bacterial health in the compost. However, it also tells us that it was not fully digested. So there were still pieces of mulch in there. And then the next two pictures are the compost divided into the cheesecloth and then being cooked in an Instant Pot. When we aerated these composts, we did it for 24 hours, and it created this interesting frothy water covering, which you don't normally see in hydroponics. So data collection, all data was taken in this chart here, and data was taken every 5 days. And the data that was taken was the height of the plants, the number of leaves, the TDS or total dissolved solids that were in the water, the temperature, and the acidity. And then using some of those numbers, we're able to produce a leaf rate, which is the number of leaves grown per day, as well as weight change and growth rate, which is how much height is added per day of the plant. So now into results. So perhaps one of the more compelling results that we saw was for transplant rates. Transplant rates refer to the number of successful transplants from the seeding tray and the humidity dome into the hydroponic solution. Generally, you can expect about a 25% transplant rate, and with our control, we got 32%. However, as you can see with our sterilized compost nutrient solution, we doubled that transplant rate and tripled it with non-sterilized compost nutrient solution. So those are wonderful findings, as generally in hydroponics, in those little starter cubes, you put 5 seeds, 1 that you assume will not germinate. So you have 4 germinating, and then you assume 3 die when you transplant. So this is really interesting because it would allow us to cut down on the number of seeds that we use in these systems. Next is average leaves per day per plant. One thing to note is that we did see in between day 10 and 15, we saw widespread mortality throughout the plants in the system. Some of that was due to acidity changes, which we'll talk about in a bit, but we do believe that this was a result of too high or too low ambient temperature in the apartment, and just wasn't something that I was able to control in the setting enough. However, this is about what we would expect for plants going from 2 leaves to 4 leaves. And this does exclude mortality, so we aren't adding in any of those plants that died off, which is why we have those nice clean numbers here. Average plant height per day. This is a very standard very standard trend that you'd see with plants, where they start off a little slower, speed up before leveling off once they've been in the system for a while. As you can see, the control didn't have, was entirely dead by day 15. Sterilized compost nutrients was by day 20. However, non-sterilized compost nutrients did have plants that continued living throughout the end of this experiment. and got up to 5 centimeters during the 30-day experiment. So growth rate per day looks at how much height was added per plant per day. And it was very similar for control and non-sterilized compost nutrient solution. The sterilized compost nutrient solution was oddly linear throughout this day 5 through 15, which is quite interesting. So total dissolved solids is a measurement of how many nutrients are dissolved within the water. Up top, we have control totes, which had the 7 tablespoons of MaxiGrow, and they were almost 1,400 ppm, or parts per million, whereas the compost solutions were all below 400. So the question that this raises is, if this experiment wasn't just 30 days long, but long enough to where we could see the entire growth of the plant, would it be enough to promote healthy and sustained growth the entire time, or would we need to supplement in more compost nutrients, or possibly change our ratio of leaching compost from at a 1:5 volumetric ratio, increasing that 2:5, 3:5, so on and so forth, to be able to increase that TDS. It's also possible that given the large amount of wood and mulch chunks that were in the compost, that having a compost that was better digested would be able to increase that number. So this is a comparison of pH, or acidity, in the water. And this is definitely one of the more interesting findings that we have here. Deep water culture hydroponics is notorious for having unstable pHs that can vary and all over the place. So here we have our controls, and as you can see, they start beneath neutral, and then we adjust those and bring those into the range that we'd like, of 5.4 to 5.9, using buffering chemicals, which raise or lower the pH. One thing that is interesting is, even though these totes all receive the same amount of water and same amount of nutrients, chemicals, they had a very wide range of acidity right off the bat. So as you can see, whenever a data point is outside the range that we'd like, it's immediately adjusted, and the new acidity level is recorded on the next day of the experiment. So we're able to put those data points in. So this is very common, frequently adjusting those pHs. However, what's interesting about the sterilized nutrient solution is that it starts a little higher than the pH of control totes. However, we have these massive swings in pH where they're dropping down to 3.2 to 3.4, or sorry, 3.2 to 4.4 pH. And both of these days we had mortality in those individual totes of the plant. So we aren't sure what caused this massive pH swing, whether that be the lack of organisms in there, or the fact that when we baked it, we actually changed the chemical composition of the compost, and then when we leached it, that of course affected the water. Now on to variable 2 with the pH of non-sterilized nutrient solution, and you can see that this starts significantly higher than the pH of variable 1. So that's something interesting to look at, what caused that, again, whether that's bacterial life or physically changing the composition of the compost through pasteurization. However, what's great here is, even though deep water culture hydroponics is considered to have very variable pH, the pH was adjusted once to get into the range that we liked it, wanted it to be for the basil, and then it maintained that pH very steadily the whole time, and no chemicals were added. So that is absolutely a benefit to using this that we don't normally see in hydroponics. So now onto the discussion. So the scalability of this experiment, it would definitely be nice to run this experiment on a larger scale. Given that we only had 36 plants here, it didn't give much room for plants dying off, which unfortunately we saw quite a lot of. This is absolutely scalable though. Compost is incredibly easy to produce at the household, local, and commercial level, as well as hydroponics, which is incredibly common for home growers, as well as commercially, where much of our lettuce, tomato, and strawberry supply in the United States comes from hydroponics. And the leaching process also would be incredibly easy to do if you had a large enough oven in a vented area to sterilize it and then leach it. So looking at future research, some things that would be very interesting to look at are other crops. Basil is a very low nutrient intensive crop, meaning that it doesn't need much to grow. However, if we wanted to look at something such as rosemary, which isn't going to be much more energy intensive given that it is still an herb, but it has a woody stalk, so it does require more nutrients. But then also looking at things like tomatoes, cucumbers, strawberries, lettuces, which are all very frequently grown hydroponically, be very interesting to see if they could sustain a full growth cycle in this compost nutrient solution. Another thing to look at would be compost quantities. So the compost that we had, we leached for 24 hours. However, every hour after leaching, I did measure the total dissolved solids, and it never, after 3 hours, it was at its maximum. So it's possible that instead of leaching for 24 hours, we could leach for just 3 hours, somewhere in that range. So comparing that would be very helpful. Next is compost selection. So as I said earlier, the compost was not ideal. Even though we knew that it had excellent bacterial health, we aren't sure of it could have had more nutrients involved, and we know that because it wasn't fully digested and composted yet. So finding a compost that has fully gone through the composting process would be very helpful. And getting one that we knew didn't have that same amount of wood chips and lumber and mulch in there would be incredibly helpful, as well as things like housework scraps from coffee, peppers, onions, your standard, what you produce in your kitchen is more dense in nutrients. Another really helpful thing would to have our compost evaluated for that NPK ratio. This would allow us to compare it to things like the Maxigro solution for more of a one-to-one comparison. What would also be great about that is if we could compare the NPK ratio of the compost before and after it was leached, as doing this would allow us to figure out how much nutrients were leached successfully into the hydroponic nutrient solution. It would also tell us how much is left over, because in this process, we don't know what we're leaving in the soil. It could be that we're creating a gallon of soil waste because there's no nutrients left over, and therefore we can't use it to plant with in the future. Or there could still be a significant amount of nutrients left over, in which case we could absolutely substitute it with more compost for traditional agriculture. So now looking at nutrient and energy inputs. One thing that's really important when growing in this situation is using rock wool. Rock wool is that cotton candy-like extruded rock that I was pointing out earlier that holds the seed and maintains the plant growing upright during the growing process. There's multiple different mediums that you can use for hydroponics. However, most of them have some sort of embedded nutrients in them. So things like peat pellets, which are effectively soil in a little screen. That's generally what I use because it's not energy intensive to make, whereas rock wool is very energy intensive. However, for the sake of this experiment, rock wool does not have any nutrients. It doesn't have an NPK ratio. So the plants are forced to pull nutrients from the compost or water solution or much earlier on compared to living off the nutrients that are provided to them in the peat pellets. Another very helpful thing would have a grow tent. A grow tent is effectively a big mylar dome that you put over your plants. and it helps reflect more light from the grow lights to the plants, as well as it allows you more temperature control and helps reduce disease and pathogen in the plants. Having this would be fantastic, as we could increase the lighting interval that the plants receive, and we could also help maintain that ambient temperature a little bit higher with the grow tent. So, some conclusions. The experiment needs to be run again. The widespread mortality, which was likely due to the high ambient temperature, doesn't allow us to make really conclusive results out of this. So, looking towards the future, we need a warmer ambient temperature. Given that this took place in an apartment with baseboard heating, the temperature fluctuated a lot. So doing this in a place where there's more control over that would be fantastic. Another really helpful thing would be to allow the plants to mature longer inside of the seeding tray and in the humidity dome, as this allows the plants to be significantly more resilient once they're transplanted into the system. As I said earlier, grow tents and lighting would be fantastic. We'd be able to grow plants quicker and be able to grow healthier ones if we increase the lighting. Also, continuous pH monitoring. That's something that can get expensive to monitor continuously. However, large-scale hydroponic farms do have this to prevent those wild swings that we saw with the variable 1 non-sterilized compost solution. A substitution to this, if this were to be run again on a small scale, would just be to measure that pH once every day instead of once every 5 days. So some takeaways. We had a 3 times higher transplant rate with the non-sterilized compost solution. We had drastically higher control over pH, given that the solution was able to buffer itself. And we had more resilient plants, which we saw given the widespread mortality in the control and throughout most of the non-sterilized, through the sterilized compost solution. So these aren't definitive results. We need to test more to be able to make these statements widespread. However, they are takeaways that we can look to confirm in further experiments. So before getting into some questions, I'd like to thank some people. First off, I'd like to thank Dr. Dahl, who was my chair before leaving for Seattle and who began this process and really got me interested in all of this. Dr. Farrell for chairing my thesis committee this semester, as well as jumping in halfway through and just really being an amazing resource and mentor through this. Also would like to thank OSR for funding this and Ms. Painter in the front office for ordering all of those supplies. I'd also like to thank all my family and friends and those that are on the call now for supporting me through this and encouraging me through this process. And now I'd love to field some questions from you all. So if you guys have questions and would like to bring yourself off mute to ask this, I'd appreciate that.
Well, Daniel, I was just going to say this is a wonderful project, and I was just emailing Dr. Dahl about something else, and I think she would love to hear the recording. So if you're willing to forward me the recording, I'll send it along to her, and I think that would make her really happy.
Absolutely. Yeah, I've been in touch with her since she's left. Definitely miss seeing her around app, but I will for sure make sure this gets to her.
Yeah. Oh, yeah. If you're in touch with her yourself, please go ahead. I think that would make her really happy.
Absolutely.
And I actually am the end.
Daniel,
Oh wait, is Dr. Dahl actually here?
Yeah, actually I
Oh,
was.
that makes me so
hi,
happy.
Susan.
Hi everybody.
How are you, Dr. Dahl?
Good.
Oh my goodness.
I know. Hi guys from Seattle. I was about 10 minutes late, but Gabby, because I didn't have the Zoom, my App State account died when I left, so it didn't get to me, Daniel. But Gabby touched base, and so yeah, I'm here. I've been here since 1:10. But anyway, I don't want to derail Daniel's day, but it's totally awesome to hear all this and that you've gotten to this point. I'm gonna let sort of the official folks go with their questions. I might have a couple myself, but anyway, hello everybody. It's delightful to see the faces that I can see. Who can I see? I can see Chip and I can see Heather. so I'll let things proceed. Hi Chip, I can see you waving. So yeah, hi, and fantastic, Daniel. I'm probably going to have a few questions, but like I said, I'll turn it back over to your committee and the other people, and I'll chime in with questions a little bit later.
Thank you so much. Really glad you can make it today. That means a lot.
Yeah, I'm totally excited.
Daniel, before we go any further, I wanted to take a moment to introduce our two guests from the Honors College. First of all, Dr. Jeff Valbush, who is the Dean of the Honors College, and of course, Dr. Heather Waldrop, who is the Associate Director of the Honors College. So thank you both for taking time out of what I imagine is a very busy week to join us for Daniel's thesis defense. And Daniel, awesome job. Thank you.
Thank you.
Awesome job. I'll just repeat that, Daniel. Really happy to be here.
Hey, Daniel.
Yes.
Hey, it's Philip. So I had a question. Obviously, these are consumable plants. Did you compare the flavor? Obviously, it's very subjective, but obviously the goal is to eat these. Did you compare the flavor of them?
So I would like to, however, given the widespread mortality and given the short nature of the experiment, we didn't have, I guess a viable plant to consume at the end of it. However, with that being said, basil is a plant that's very commonly grown hydroponically, as well as lettuces, tomatoes, cucumbers, strawberries are mostly grown hydroponically in the consumer market today. So this is something that's happening and increasing every year, and most of the consumer market doesn't know that.
All right, so you all know me, and I can't sit here quietly with bursting with questions. And I'm sorry I came in a few minutes late, but one of the things that struck me is one of the first things that I heard was that that the curve that showed the number of leaves and the fact that your plants that had either sterilized or not sterilized compost died, whereas your sterilized compost continued to grow. What was that all about?
So I believe the widespread mortality, and there's so many factors that could have gone into this, but I think the most reasonable thing that I could pinpoint would be temperature. Basil's a warm weather plant, and the past 45 days in Boone when I was growing have not been warm weather. So that really was something I probably should have changed the plants, hindsight being 2020. But so I think temperature was the largest thing to blame there. And it's possible that the sterilized compost or the non-sterilized compost solution, both of those provided a more resilient plant somehow. It's also possible that it's a fluke genetic thing, and we just need to do more testing with more plants to be able to make a definitive call there. Thank you.
So Daniel, you've done a lot of work behind the scenes here. I was encouraging him to show images of the project, but he said, "No, you don't want to see my living room." So this was in his apartment living room over months and months, and I got to hear the updates, both the plant growing and the plant dying. But I think there were some really good insights that came out of this. And so my question is, the next time you do this, because I imagine you'll do it again in the future.
Absolutely.
What are the main things you'll do differently?
So absolutely taking pH more often. The reason why we did every 5 days was I grow a bunch of food and stuff hydroponically. And when I was just kind of estimating at home, every 5 days was when I could see a difference in the height, a measurable difference in the height of a plant. So that's what I decided to check all the variables on, was every 5 days. So checking pH every day, making sure that it's within that 5.4 to 5.9 range, it's absolutely something that would change. Increasing the lighting factor by bringing the lights closer to the plant, as well as providing more lights, would absolutely be very helpful. So I'd love to run it with basil again because I love growing basil. It's so versatile, but then eventually moving on to other plants and seeing if it, like I said, it can support a more energy intensive plant.
And you think you could use cuttings or clones of basil to remove the question of genetic diversity?
So that's something that we talked a lot about during our weekly meetings, was the genetic variability, given that everything started from seed. The actual the species of basil that we used was very specific, meant for growing hydroponically in tight quarters. However, there's still different seeds with different genetic qualities. So a thought would be to propagate basil so that we have 36 different basil propagations at about the same stage of growth. So then we have a more resilient plant and the exact same genetic variability throughout the plant. So then we can rule out genetic variability almost completely from the experiment. Doing that would take quite a lot of time, just given that we'd have to start from one basil plant, allow it to become mature enough to where we could propagate, and then do that 36 times. And I did the math for that. It would take a while. It's definitely something that I would like to do, but that's definitely something that would occur year and a half, 2 years down the line in order to get 36 propagations at the same stage of growth.
Right.
Again, sorry for coming late, but I missed, where did you get your compost? And do you have any thoughts about the composition of the compost and how that might have some effect?
Absolutely. So the compost that was collected was compost that's produced by the physical plant and dining services. And the compost was not ideal. It would definitely be great to test again with a different compost composition, because the one that we received was about 130 degrees. So that means that we have excellent bacterial health and growth, but we don't have a fully digested compost, as well as it had a large presence of mulch in there, which is hard to leach from. It would be better if we had a well-digested soil compost like instead of having those wood chips in there.
Mm-hmm.
Daniel, I have a question about electricity. I understand the drawback of hydroponics is how much electricity it used, that's what I understand. And I wonder how much your electric bill went up when you were doing this project.
Yes, so it's surprisingly minimal. So this was 36 plants. I cannot speak to the heating cost of the water. I know that those were 50-watt heaters. However, it's hard to gauge when they were turning on because they were set to a certain temperature. So they'd reach that temperature, turn off, so on and so forth. However, combination of the lights, so we had two 24-watt lights that ran, was 11 hours a day. And then the pumps, I forget the exact wattage on those, but for this month was just under $1 of electricity. And we were using, the totes that we were using were very small. I generally would run, so we used one pump for 4 plants. Generally in my home growing system, I'll use one pump for 12 to 16 plants. So that's absolutely like an economy of scale. The more plants you have, the more you can drop that cost per plant.
And that could be an easy thing to monitor, just stick a kilowatt meter. If it was
Absolutely
important, that could be easily added.
Mm-hmm. Especially if we just like running this again, getting that and just running our, they aren't cheap, but just running our heaters off of that, because we can easily calculate everything else off of the wattage and the amount of time that they run. But being able to know those heaters would be incredibly valuable.
Daniel, I have a question. Great job today.
Thank you. I appreciate it.
As you may know, I'm the director of the graduate program. So my question is, have you applied to graduate school yet?
Not yet. So kind of making a crazy 180. I'm about to wrap up my EMT through Caldwell. I've been spending 10 hours a week there this semester getting my EMT, and then plan on working as an EMT for a year or two as I do my prereqs for nursing school. So definitely a bit of a funny 180, but I'm looking forward to it.
That sounds great. Good job.
Thank you.
Dan, I just want to say to show off your expert status among your group of friends, to all of your mentors around here, that next time you come over before a bike ride or something, I need to get your consulting work on the gardening habit that I've acquired.
Sounds good. I'd be happy to consult.
Everything I'm doing and all the best practices. Yeah, you spent some time studying something that's now kind of zeitgeisty, so that's kind of cool.
I'd love to help.
That's right. This is a perpetual interest among students, and everyone, I feel like, is doing these small scale hydroponic systems that are really fascinating and interesting. And Daniel's really larger question is exposing some of the footprint, the carbon footprint of like Heather's questions about electricity and energy use, but also about the rock wool and the media itself, and importing nutrients. That's not necessarily the best solution. And if nutrients can be made locally, then and reduce all that transportation cost and refining cost, it has a real potential here. So I think this is a really meaningful area, and also a fun hobby and background. I'm sure you'll always be growing plants.
Absolutely.
So yeah, you've done a very thorough job. It's been a pleasure working with you, and I was happy to be a part of this group. And I'm glad that Dr. Dahl is here, who got this started off. So yeah, thanks for your diligence and your hard work on this, Daniel. You've done a great job.
Thank you.
And I just wanted to put in a couple words about on Jeremy's that there are lots of questions out there, and there's lots of like closed system questions, which is obviously part of my work now. And there's a lot of even terrestrial like container type things going on. So I think you've really laid some good groundwork here for the kinds of questions that could be really important for those sort of more closed system, and can we do things in a more holistic sort of recyclable way. I think it's really important work, and hopefully there will be other students that'll follow in your footsteps.
Thank you. I appreciate it.
Sorry, I interrupted after Jeremy's. You're doing a great job.
Thank you. I appreciate it. And I appreciate everyone coming on, and that my whole team was able to make it. Really appreciate your all's support and advice over the past year or so of getting this process all set up and going through the motions of the experiment and writing it. I really cannot thank you guys enough for that.
And thanks for everybody for joining in. I think the attendance actually may be better than an in-person defense.
Yes.
So this is really nice. Thanks to the family members and everybody.
Thanks, everybody. Stay safe.
All right. Thanks. Daniel will be in touch about the last few steps. And yeah, great job again. And thanks, everybody, for joining us on this defense.
Thank you. Thanks, Joe.
Thank you.
Thank you. Congratulations, Daniel.
Thank you very much, everyone. I appreciate it.
Bye-bye.
Daniel.
Way to go, Daniel.
Daniel, I think I'll have the recording, so I'll make sure that I send that to you.
Excellent. I appreciate that.
Susan's still here.
Yep, I'm still here. Hi, y'all, whoever still left.
How are you doing, Dr. Dahl?
I'm actually doing great, although it's pretty weird because I've been working from home for like more than half of the time I've been here.
That is strange.
Right. That's right.
So it's a little weird to get to used to a new location and a new job when I'm sitting here by myself in my apartment. So, but it's really exciting, and this is just reminding me of like the kinds of stuff that y'all are doing, because one of the areas that obviously I'm looking at and responsible for is kind of the long-term objective of like growing food in space. And so, I mean, I'd love to have a chat. I don't know, however you would want to arrange that, Jeremy, just in terms of some of the things that I've been thinking about it in kind of the long-term
Mhm.
perspective of some things that we really need to know in terms of how to do that.
Mhm.
And you really touched on it perfectly. There's lots of little things that we, really need to know more about. And I think Daniel's sort of closed the loop with, the compost leachate is a perfectly good example of how might you want to do that in an environment where you really are trying to recycle things. So,
Mhm.
anyway, so I'm having, as you can probably tell, I'm having a really great time thinking about all this really wide-ranging stuff. My job is sort of like, wow, really? You get paid for that?
So great. Well, you deserve it more than anybody. It's really glad to hear that.
Yeah.
And yes, be happy to touch base with you. And I think, some of Daniel's findings about this, the transplant success ratio and the pH buffering, and some of these effects and how to even make an appropriate compost tea based on TDS. Daniel, that was a good find, I thought.
Absolutely.
So relatively inexpensive to do that. Use TDS meters or even pH. But there's some really interesting things that have come out of this that can. I know there'll be another student.
Yes.
Right away. I mean,
Well, make sure to point in my direction. I'd love to talk to him.
So your thesis will serve as a good resource for that.
Thank you. I appreciate that. And it's in the drive, the final version, as well as all the numbers and PowerPoint.
So Daniel, what's the timeline? You've obviously got to get this finished up, and graduation is kind of in there somewhere. However that's going to happen. I've been thinking about you all trying to do what you do from home. And then when do you start your EMT job?
So I actually am unfortunately having to miss virtual graduation for EMT stuff now. We've got our ride time that day. So, but I'll test for this on the state exam, I believe on the 19th of May, and then test for the national exam, I think the 22nd it is. And then I'm looking to get hired in one of the counties up here where I have connections before moving either back to Atlanta to take prereqs, or I'm looking to move to Raleigh possibly, and then getting tied in with either Duke or UNC's critical care team there. So lots up in the air, but I'm excited for it all.
Yeah, that sounds great. You've got a plan, and you just kind of have to see how it unfolds. So.
Yes, absolutely.
Hey, Susan, while I have you here, can you give me your new email or your preferred email?
Yeah, and anybody else who wants it, you can.
I sent you an email a little bit ago, and I don't think it went through, so I'm going to write that down as well.
Yeah, it's SCDOLL2, the number, at gmail.com. And my phone is the same, so those are the two numbers. My laptop doesn't have good Zoom or Skype, but I have a work laptop that has all of those things, so I can participate, via it.
Great. Yeah, we're finally using the full functionality of AppState's Zoom license.
Oh, good. Well, I mean, and that's the other thing that I'm finding, and maybe y'all can too, the fact that there was good attendance. I think in the future it would really be good to allow that as an option.
Yeah.
We've found from working from home, a lot of what I do, I can do from home, but we've actually gotten more efficient by using Skype chat and Skype calls, and because I used to have to walk across the full room to get to see if my boss was at his desk.
Right.
And I can just send him a chat, and if he's there, he answers it and things. So I think that's really well worth sort of looking into what have we learned that actually might help us in the future. And the fact that there were so many people able to attend, I think that's awesome. We've always had sort of low turnout. And I think that that's always been unfortunate, because I think that seeing students in their research is a really fantastic thing. So.
Absolutely. And it allows someone like you to join from the other side of the country. I'm 3 time zones away. I mean, I had 2 people from your time zone join, and that's awesome. Like,
Yeah, yeah. I mean, and that really is, and because I mean, I'm certainly, invested enough and interested enough in all the things that are happening that I would love to be on a continued distribution list. Jeremy, or I don't know if Jeff is still with us, but even grad student thesis like that.
That's great.
If they're available, via Zoom, I think it'd just be a really great way to sort of stay connected.
Absolutely. Yeah, it's really, this is a really unique opportunity to gain a lot of things that we're learning and implement them going forward. I mean, in some ways, I don't think things will be the same. We're always going to be working a little differently now.
Yeah, no, I totally agree with that. And I just sort of have to laugh because both you and Jeff have these awesome backgrounds. How do you do that?
There's a background option. Choose the virtual background. So I don't have any.
Because the Northern Lights is pretty awesome. Oh, there we go. Okay.
This is a university official background.
Okay.
Or this one.
Add that every time we had a meeting, like before I'd log on, I'd be like, I wonder what background Dr. Farrell's going to have today. Like, it was part of the fun of it. Like,
and there's this one
background.
Oh, that's even better.
I like that.
Although it is a little weird because you have like this paper cutout outline around your head.
Right.
So
it's better than my guest bedroom, which is this.
Oh. Okay, there. Yeah, I'm sitting in front of my china cabinet, so it's pretty okay. But yeah, it's sort of weird to be talking to colleagues and you see like their bed in the background. Or we had my boss's boss had an all hands meeting, and his cat was climbing around on the bookshelf behind him during the call.
I love that.
Yeah, so that was pretty interesting. But it's also been interesting because I don't have human contact here, so that alone by yourself in the apartment is getting a little bit old. But my colleague, my boss has organized like a half-hour coffee hour, and so people will be sitting in their backyards or sitting in interesting places. So it's like, whoa, just these little glimpses into people's lives that when you just see them at the office that you don't really see. So that was pretty interesting. But anyway, yeah. So everybody doing okay on that front in terms of the working from home? And
I think so. I mean, I don't know, Daniel, but I think most of the students are ready to be finished. It's just been hard to stay on track and be motivated. And in my classes today, they said, the workload's down a little bit, but it's so hard to be motivated. And this is the crunch week. And anyway.
I've only heard from maybe one or two students, and they're just like, me, the motivation thing is just so hard. It's just so hard to
the library's not open, and all the places that you're used to going to do your work are not available. And so you have to now work from home in a place that you used to just, relax. And and now you're having to really, it's kind of a good lesson in discipline for getting out, or doing a thesis, Daniel, or doing something like that where it's all intrinsic. But
absolutely,
it's been tough. I think it's been tough on anybody, but the students are vocalizing that now.
Yeah. Well, hopefully the whomever will get their act together and get a vaccine in the works, at least. I was listening to something just the other day. Bill Gates was talking about, he's actually helping to fund, I think, 7 different actual production facilities. He said they're actually developing them in parallel. They don't know which of the 7 is actually going to have the one that they eventually settle on, he said, but at least if whichever one they do will be up ready to be produced. Wow. So he was saying the money that was going to be wasted on the other 5 or so that weren't actually going to go to production, he said, well, that's small change compared to every month that goes by with the economy in the tank.
Right.
So anyway, that was just really interesting that there are efforts going on. It may not be government led, but they're definitely trying to do that. And that's kind of where I'm standing, given my age and my health status, being in a high risk group, that I can work from home and probably will be until there's a vaccination. Because there's no reason for me to take the risk until they do. So, and there's scary stuff coming out about all the other things that are going on besides this just being a flu-like respiratory virus. That people should be really, really scared. Clotting and brain, strokes and other things associated with
Now
the
with strokes now is interesting.
bizarre. Yeah.
Wow.
So
Daniel,
anyway,
it's a big testament to the profession that you're going into. So this is
Yeah, I'm a little nervous, but I'm excited at the same time.
yeah, no, it's really laudable. I think that's going to be awesome. And talk about making a difference and immediate.
Yeah, no, every day that I'm not out there, we were supposed to have ride time, and that got pushed back. And it's like I just want to be out there working on this,
Yeah, yeah.
So.
Yeah, so that's really great. All right, well, it's still the middle of my workday.
Oh, yes, it's 1 year time, I guess.
It's almost 2. So anyway, super great, seeing both of you. And do please, Jeremy, feel free to pass on my contact information. And if there's ever you or a small group or whoever that wants to talk about, just things and questions that could be very relevant for closed system agriculture, I'd be very delighted to participate.
That sounds great. Yeah, I've got your updated email. I've got your phone number, your cell phone number still. Yeah. So let's get together sometime and just have a little coffee hour over that.
Oh, absolutely. That sounds really great. Yeah, my days are pretty much blur together. So I know summertime, you've got kind of the peach and the push coming on for the end of the semester, but so whenever it fits your schedule,
okay.
Just give me a call. We'll work something out. So.
All right, Daniel, well done. Well done. I think since I scheduled the meeting, I'll have a recording and I'll make sure I send you the link and the MP4
I'm going to sign up, guys. Bye.
Oh,
bye, Susan. Good to see you.
Wait, just a second.
Oh, wait.
Dr. Waldrop wanted a picture. So I'm going to take a selfie with you guys to send to her for
okay.
the website.
Selfie, huh?
Yeah.
How does that work?
Just like this.
You got us both in the picture?
I think it's a little blurry. Let's see. Yeah.
The infamous spring 2020 semester.
I know. It'll go down in history books. Okay, hopefully this turns out well.
All right, that's awesome.
I'll send it to you guys as well.
Cool. Okay. Thanks so much, Daniel. And thanks, Jeremy, for this. This has been really great.
You're welcome. Good to see you.
Bye. Congratulations.
Thank you.
Bye.
Bye.